Good day, everyone. Welcome to today's virtual lesson on Meiosis. I'm Marinel Baldo, together with Loida and Ronalyn Budkan, your instructors for this session. I hope you're all ready to dive into some exciting learning. So before we proceed with our lesson, let us take on this little challenge where I will flash four pictures and you will guess what it is trying to convey. Number one, we have a sperm cell and egg cell and plant and animal cell. Any guess so far? What do you think is common between these four? Yes, you're right. The answer is cell. What about the next item? We have a picture of a man, a gender, and a signage. What do you think is the answer? Man is also known as a. Yes, indeed, it is a male. I knew the next item will be pretty easy for you to guess. The answer is what? Yes, you're right. It's a female. Based from the previous slides, what now do you think is our lesson for today? With the clues from your answers, any ideas? None? Let's take on this very last item. Maybe we could have some ideas. So, what does the cat say? And what do you think is the relationship of these two females? Yes, sisters. So, from the given clause such as cells, female, and male, our lesson today is on the cell division, meiosis. So, our topic is on meiosis. But before we start, does anyone know who this guy is? So, he is Oscar Hertwig. He is accredited as the first person to directly observe sexual reproduction using a microscope. He studied sea urchins and witnessed the fusion of sperm and egg cells during fertilization. So, what now is meiosis? So, meiosis is a type of cell division where a single cell divides twice and produces four daughter cells with half the amount of genetic materials.
Now, let's look at the stages of meiosis. So, meiosis is the reproductive cell division that occurs in the gonads and produces gametes. So, the first meiotic division is complex and involves the following phases. First up is we have the interphase. The interphase is the very first phase of cell division before it will proceed to the meiosis phase. So, interphase involves the growth phase 1, wherein the cell will grow and attain its normal size. So in here, organelles will be formed and proteins such as enzymes needed in the S phase will be produced. So after growth 1 phase, it will now move on to is S phase or the synthesis phase, wherein the duplication of the DNA occurs through the process called replication. So after the S phase, it will now move on to growth 2 phase, wherein the cell will check if there are any errors in the replication of the cells or of the chromosomes. So, after that, it will now move on to meiosis. And the first phase in meiosis is the prophase 1. Prophase 1 is known as the longest phase that occur in meiosis because it involves 5 stages. So in prophase 1, this is where the chromosomes will condense and become visible. the homologous chromosomes, or the chromosomes that code for the same traits, will pair up with each other, and a process called crossing over occurs where they exchange genetic material. So, the 5 stages that occur in prophase 1 are: number 1 is liptotin. Liptotin is known as the bouquet stage, wherein the chromosomes will condense. Next up is the zygote, where 2 pairs of homologous chromosome will pair up. And we have the pachytene, where compaction occurs, and the compaction will result in exchange in genetic material, or what is known as the crossing over. Next to pachytene is the diplotin, where the homologous chromosomes will separate. And after diplotin is the diakinesis, or the total separation of the homologous chromosomes. So overall, after these 5 stages, it will now move on to metaphase 1. Metaphase 1 is known wherein the chromosomes will align in pairs at the center of the cell. The center of the cell is known as the metaphase plate. And then after that, the spindle fibers from the centrosomes will connect to the centromere of the chromosomes. Then after that, it will now move on to the process anaphase 1, wherein the chromosome pairs separate and move to opposite ends of the cell because of the shortening of the spindle fibers. In here is where the reduction in chromosome number occurs in preparation for telophase 1. So, a cleavage furrow will form, and the nuclear envelope will form around each set of chromosomes as the cytoplasm divides, or what is known as the cytokinesis, resulting into 2 daughter cells. However, unlike mitosis, these daughter cells each have a full set of replicated chromosomes which are still diploid. Diploid means a set of cell that has a complete set of chromosomes with a total of 46 chromosomes. But the chromosomes themselves are no longer identical due to the crossing over that occurred in the prophase 1. So, after telophase 1, it will now move on to the second process, which is the meiosis 2.
Okay, moving on to the next stage of meiosis, which is the meiosis 2, wherein DNA does not replicate in this stage. Meiosis 2 has also primary rule, and that is to separate the sister chromatids that was produced in meiosis 1. There are 4 phases of meiosis 2, and these are prophase 2, metaphase 2, anaphase 2, and lastly the telophase 2. But first, we will discuss the prophase 2, wherein the chromosomes condense and the nuclear envelope breaks down. After that, it will move to metaphase 2, wherein each chromosomes will attach at a special point called centromere or in the metaplate. After that, the centromeres split, separating the sister chromatids, moving them to the opposite ends of the cell. And this will ensure that the daughter cell remains haploid. That means that they have the genetic material of the original parent cell. And lastly, it will end to telophase 2. In this phase, a nuclear envelope forms around the set of chromosomes, and then the cytoplasm divides. That will result in 4 daughter cells. And they are genetically unique due to the random separation of sister chromatids.
So now, let us review on the whole process that occurs in meiosis. So it is said that we have meiosis 1 and meiosis 2. The first process that will occur in the cell division is interphase, wherein it will undergo growth 1 phase, synthesis phase, and growth 2 phase. After that, it will move to prophase 1, where the chromosomes will condense and the centrosomes will move to the opposite ends of the cell. Next up, it will move to metaphase 1. Metaphase means in the middle. That's where the that's where the chromosomes will align in the metaphase plate, and the spindle fibers will connect to the centromere of the chromosomes. After that, it will move to anaphase 1. Anaphase 1 is the moving away of the chromosomes to the opposite side of the cell. Next up is in the telophase 1, where it is characterized by the occurrence of the cleavage furrow and the separation of the cells, the formation of the nuclear membrane, and the nucleolus will appear. The total separation of the cell or the daughter cell is called the cytokinesis. So after telophase 1, it has produced 2 daughter cells. So the 2 daughter cells are diploid and will now move to the second process, which is the meiosis 2. The 2 daughter cells will then repeat the process again, which is which will start in prophase 2. Next up, it will go to metaphase 2 and move to anaphase 2 and then telophase 2. So in telophase 2, cytokinesis will occur and then these cells will divide into 2, resulting into 4 granddaughter cells. So the 4 granddaughter cells are haploid. That means the chromosomes are divided in and are unlike the daughter cells. So, any questions? Have you get the whole process so far? One important aspect we should know right now after learning about meiosis is its significance to our life. So meiosis makes sure that all organisms produced via sexual reproduction, which includes us humans, contain the correct number of chromosomes have from each parent. This explains why our genetic traits are different from our parents.
Now, let's see how far you're understanding with a few quick evaluation questions. I'll ask the question and I want you to pause the video to think about your answer. Are you ready? And 3, 2, 1. Here is the first question. How many daughter cells are produced in meiosis II? Again, how many daughter cells are produced in meiosis II? Second item, who first observed and described the process of meiosis? Who first observed and described the process of meiosis? The answer is Oskar Hertwig. He is the one who first discovered the process of meiosis. Number 3. It is also known as metaplate. Again, it is also known as metaplate. The answer is equatorial plate. Next, it is where the spindle fiber attached to the homologous chromosomes. It is where the spindle fiber attached to the homologous chromosomes. It is kinetochore. Next, the covering that disappear during the prophase and reform around the chromosomes during telophase. Covering that disappear during prophase and perform around the chromosomes during telophase. The answer is nuclear membrane. Next, it is where the sister chromatids attach to each other. It is where the sister chromatids attach to each other. The answer is centromere. Next, what is formed during telophase phase? What is formed during telophase phase? The answer is cleavage furrow. Next question. The reproductive cell division that occurs in the gonads and produces gametes. Again, the reproductive cell division that occurs in the gonads and produces gametes. The answer is meiosis. Next question. Meiosis that begins once the chromosomal replication is complete. Meiosis that begins once the chromosomal replication is complete. The answer is meiosis 2. Next question. It is responsible for the separation of chromatids to opposite poles of the cell. Again, it is responsible for the separation of chromatids to opposite poles of the cell. The answer is spindle fiber. That's the end of our discussion, and thank you for watching and listening. Bye.